Good day, everyone. This is Jeroen den Dunnen calling in from the Netherlands, the Amsterdam UMC. Now, unfortunately, I'm not able to join this meeting live, but through this video, we would like to update you on our most recent work on autoimmunity and long COVID and ME/CFS. But before I do that, I would like to underline that long COVID is really a heterogeneous disease with different types of patients. And you can make a broad categorization into two. For example, the long COVID patients that initially, with their COVID, have been hospitalized. So the patients that had severe disease, often suffered from tissue damage. Now, this group is characterized by more males, more frequent obesity, and often comorbidities. On the other hand, there's the non-hospitalized patients that had mild disease. And this group contains way more females than males and really seems to be a different group. In my presentation, I will focus on this non-hospitalized group of long COVID patients. So for a long time, it has been known that COVID-19 itself, but also long COVID patients produce autoantibodies. One of the hallmarks of autoimmunity. And these papers we've known for a long time, and they are often also associated with specific, for example, neurological symptoms or cardiological problems, etc. But the big question is it just an association, or are these autoantibodies really causing the disease? So is it correlation or causation? And that's something we really wanted to look into in our study. So we thought, how could we test this, whether there's really a causal role for autoantibodies in long COVID? And we thought of this experiment. So we thought, okay, what if we take blood from long COVID patients and patients that recovered from a COVID-19 infection and didn't have any symptoms? If there would be autoimmunity, if we would isolate the antibodies, specifically the IgGs, the autoantibodies would be in there. And then we could transfer these antibodies to another species, for example, to mice. And we cannot give questionnaires to mice, but we could do a battery of behavioral tests to see if the mice would develop symptoms. Now, we didn't just think of this. There is a precedent for this in another disease, fibromyalgia. And in this beautiful paper from the Swenson and Anderson group, they transferred antibodies from fibromyalgia patients to mice, and the mice developed symptoms. So still, there's 100 ways in which an experiment like this could have gone wrong, because you go from humans, do they have the same proteins, do they have the same antibodies, is there enough cross-reactivity, etc. But we thought, let's just do it. So that's what we did. We took two groups. So we took, of course, controls, non-long COVID patients and long COVID or PASC patients. And we did like a serum analysis to see if there would be any differences that we would observe there. So we checked some pro-inflammatory cytokines, and in the PASC group, long COVID group, IL-1 beta was a bit higher in GM-CSF. Also, the type 1 interferons were elevated, while the type 2 interferons were actually lower. And also, we saw increased neuronal damage markers in the long COVID population. Now, we did some more digging into the phenotype of these patients, and based on that, on both serum analysis with conventional methods and, for example, Olink analysis, we categorized the 34 long COVID patients into three groups based on this serum analysis. And we color-coded them, so it's gray, yellow, and red. And this is kind of the overview of these different categories. So gray showed increased type 1 interferons, red showed increased neuronal damage markers and pro-inflammatory cytokines, while the yellow group didn't really show any major elevation markers in there. And then, of course, we started doing these experiments. So taking the different categories, purifying IgG, intraperitoneal injection, and then the behavioral tests. We did a whole range of behavioral tests, pain-associated, motor activity, motivation, and cognition. I'm not going to show them all of them to you. I'm just going to show a few highlights that were quite interesting. So let's start with mobility. So we did a mobility test, a very clear and simple open field test, just 24/7 analysis of how the mice are moving all this time. We followed them over a period of about 2 weeks, as indicated here on the x-axis. On the y-axis, you see here how much they move. In black is indicated the controls, and what you can see immediately is that the yellow and the red group don't really show major differences, but immediately we saw differences in the gray group, which moved less already at day 1. There were also some other remarkable differences in this gray group. They didn't only show reduced mobility, but also showed a lower walking speed and a tendency of reduced muscle weight in the course of 2 weeks. Another test, which was very, very striking, actually most pronounced, were the pain tests. So we did a von Frey test, which indicates pain. So you kind of poke the mouse with a stick to see when he would retract the paw. And that's indicated here. So again, black is indicated the control IgG from healthy individuals, and what you can see is severe pain symptoms in all 3 groups. Now this is a log scale, and remarkably, the gray group, which showed symptoms on mobility, showed the least amount of pain, while the yellow group showed very pronounced pain symptoms right away, and the red group kind of showed different kinetics with a delayed pain response over the course of time. If we start digging into these data, later on we also saw differences between the males and the females in these groups. Another test that we did was, for example, a rotarod to see, how quickly we could, how long it would take for the mice to fall off. We didn't see significant differences there, but it is interesting that one of the other groups, the red group, was the only one that almost showed statistically significant differences there. So, what's the take-home message from there? 3 different groups with different serum analysis and different symptoms. Take these with a grain of salt. This is really a generalization. But we saw different symptoms depending on the group. And this, to our vision, indicates like 2 things. One is that the IgG antibodies from patients actually cause long COVID symptoms. So there's really a causal role. And on top of that, that the antibodies can induce specific symptoms. So different groups of patients, the IgG can induce different types of symptoms. Because there's a huge range of symptoms in these patients, and they could actually be caused by different types of autoantibodies. And since we see this now here, and this has been done before in fibromyalgia, we're now actually looking into doing the same experiments in ME/CFS patients, Lyme disease, and perhaps even acute fever fatigue syndrome. So what can we do with these findings? And of course, well, one thing that is pretty cool, we think, is that we now have an in vivo model for long COVID. So by taking blood and taking the antibodies, we can actually give mice long COVID symptoms. Which could be very valuable to test candidate drugs that we want to test to treat long COVID. But also, since we're now just trying to identify the autoantibodies and the autoantigens that are targeted by these antibodies, we could use this as a biomarker for an objective diagnostic test for long COVID. And with that, I would like to finish this presentation by thanking all the people that were involved in this. Now, fortunately, I cannot answer questions right now, but if you have any questions, don't hesitate to contact me on my email or through my socials. Thank you.